Neurons are the cells that are involved in the three nervous system functions described previously. They are the communication cells of the nervous system. To communicate, they create electrochemical signals called action potentials that then lead to the release of chemical messengers called neurotransmitters. It is the neurotransmitters that ultimately signal and communicate with other cells. Although there are a few types of neurons in the human body, all of them have structures in common. The neuron soma, or cell body of the neuron, houses the nucleus and most of the major organelles, such as the mitochondria, the endoplasmic reticulum, the Golgi apparatus, and the ribosomes. Where neurons differ are the cellular extensions from the soma. The first of such extensions are the dendrites. These are short, branch-like structures that protrude from the neuron soma. They function to receive information from other neurons, other cells, or sensory receptors in the body. They are sensitive to neurotransmitters released by other neurons, or they are sensitive to various stimuli due to the presence of receptors for those neurotransmitters or for those stimuli. There are often hundreds of dendrites per neuron. Some neurons only have a single dendrite, and others have thousands of dendrites. Diagram 12.8 highlights the cell soma, cell membrane, and the dendrites of a typical neuron. The axon is a single, typically long extension from the neuron soma. The end of the axon is called the axon terminal, and usually the axon branches near its end to form many axon terminals. The function of the axon is to create action potentials. The axon terminal possesses an enlarged region called the synaptic end bulb. The bulb contains structures that store neurotransmitters and then release those neurotransmitters when the neuron is stimulated. They form synapses with other neurons or cells so that the neurotransmitters can serve as the chemical messengers to signal those neurons or cells. Neurons may or may not possess a structure called myelin. For the neurons that do possess it, it is a specialized nervous system cell called a glial cell that wraps the axon at regular intervals. For neurons in the central nervous system, glial cells called oligodendrocytes form myelin, and for neurons in the peripheral nervous system, glial cells called Schwann cells form myelin. In either division, myelin is a structure that wraps the axon at regular intervals and functions to allow the action potential to travel faster and further down the axon before the action potential must be regenerated. Loss of myelin can lead to conditions such as multiple sclerosis, which is an autoimmune disease where the immune system kills healthy myelin cells, which leads to muscle weakness and loss of muscle control all over the body because those neurons are unable to generate action potentials normally. There are small gaps between adjacent myelin cells called the nodes of Ranvier, which permit the action potential to regenerate at the exposed axon within the node. The neuron soma tapers at one end to form the axon, and the axon extends to its axon terminal bulbs. Typically, axons branch at their ends, forming many synaptic end bulbs. Myelin wraps the axon at regular intervals, leaving small gaps of bare axon called the nodes of Ranvier, which are exposed so that the electrical activity of the action potential can be regenerated at the nodes. For neurons of the central nervous system, myelin is formed by cells called oligodendrocytes, while neurons in the peripheral nervous system have myelin that is made from cells called Schwann cells.